This is a production of Cornell University.
Great. Well, thank you, Chris. And thanks, Chris, Bill, Gillian, for the invitation. And thank you for everyone who's taken time to meet with me in the last couple of days. It's been really an incredible experience for me, and I look forward to more interactions. So Chris already mentioned social media. Do me a favor, if you're on Twitter, take out your phone and follow me. Jim Bradeen. I think I follow half of you at least already. But I, like many here, I'm really passionate about science communication, science outreach. I'm thrilled to see so many students and faculty and staff engaged in science outreach and science communication here. I think it's really critical that scientists actually talk science beyond our academies. So follow me, I'll follow back. And if you want to live tweet, you've got my permission to do that. So I've had a lot of really great conversations while I've been here. A lot of time interacting with students, which I love. One of the conversations that we've had is, how did you as a student get into the field of plant pathology? And repeatedly, I'm hearing that it's so important that a student knows a plant pathologist, that a student has a research interaction, an internship, or takes a plant path course. So that seems like a really important way to get into this field that we really think of as a discovery field. Now, for me, my path was a little bit different, but while I've never actually studied at Cornell, Cornell's played a really prominent role in where I'm at today, in a way that's probably not very obvious. So I actually grew up on a grape farm, Concord Grape Farm in Southwest Michigan. By the time I was 10 years old, I was really an active gardener. And I started reading a magazine called Rodale's Organic Gardening. Now back then, this was, I'm dating myself here, but in the mid 80s, and organic gardening was not a thing, at least in Southwest Michigan. And I remember my father pulling me aside and saying, organic gardening doesn't work. It's something the hippies do, and don't waste your time on that. So clearly he was wrong. But I read this magazine religiously, and I learned a lot about gardening, and I learned a lot about plant science. And actually, it was the pages of organic gardening that taught me that I could actually have a career as a plant scientist. And one of the names that kept coming up over and over and over again was Henry Munger. And so, if you don't know, does anyone not know? Well, that's a rough question, right? So let me tell you, Henry Munger was an important plant breeder and a professor at Cornell University. And for some reason, he was very well featured in this magazine back in the mid-1980s. So he really, more than anyone, I think, influenced my decision to go into plant sciences. And really, this magazine and his career lit a passion in me that continues to this day. So thank you, Cornell. I've always had a soft spot for this institution because of that connection. So it's really an honor to share with you our research. I work at the plant end of plant pathology. One of the hallmarks of our work has been mining crop wild relatives or wild species related to crop plants for useful genes. And I really work in the area of the genomics of plant disease resistance. So one of my goals is to use gene bank collections to identify genes for crop improvement so that they're more resistant to pathogens. Now, for many, many years, I've worked in the potato late blight pathosystem. And if you're a plant pathologist, I'm sure you already know this story. This was the causal agent of the the Irish potato famine, or the European potato famine of the 1840s. So a plant disease coupled with social factors that made an Irish population almost wholly dependent on potato for their existence. So we had some really bad years in the 1840s, really intense late blight epidemics in potato. 1 million people starved to death in Ireland. 1 million more left the country, including the ancestors of John F. Kennedy. So quite literally, if it hadn't been for this plant disease, the US probably never would have had a president called JFK. But this disease, which was so important in the 1840s, is still one of the leading biotic constraints of potato production worldwide. The price tag is somewhere in the neighborhood of $5 to $8 billion annually. So it really is a very significant disease. So one of the efforts that my group has focused on over the years, and we're one of many labs around the world to do this, is the discovery of genetic resistance to late blight. Now, to give you a visual of just how devastating this disease can be, I'm going to share with you a month in the life of potato late blight nursery. So this is a research plot at the University of Minnesota. We don't use fungicides here at all. We intentionally release the pathogen. We use overhead irrigation to maintain high humidity. And when the pathogen's happy, a field that looks like this can turn into a field that looks like this. Now, obviously, the majority of the plants in this field, the breeding lines, the potato cultivars, are susceptible. But there are a few islands of green, such as those shown there. And more often than not, those islands of green are wild species of potato. So this is one of the species that my group has worked on over the years. So it originates, this one originated from Mexico. We got the materials from a gene bank. And through phenotypic work such as this, we're identifying resistance genes that could be incorporated into potato. Now, this approach is inherently phenotype driven. And that's slow and laborious. Now, phenotyping is a lot sexier today than it was back even a decade ago. And so my colleagues at University of Minnesota, folks here in many institutions across the globe, are working on remote imaging as a way of speeding up the plant phenotyping process. So we're taking pictures of fields with drones or with field-based apparatus, or we're doing laboratory analyses. Of course, data collection is one thing, data analysis is another. And so plant phenotyping is getting much larger scale, a lot of effort in terms of training computers to recognize what plant disease looks like, for example. So while this is a lot faster and more high throughput than walking the field and looking for those rare islands of green, this is also inherently based on plant phenotyping. So if you think about a wild population of plants, or you think about a gene bank collection, we know there are useful genes in that set, but we also know there are a lot of genes that aren't useful for crop production. So finding a useful disease resistance gene in a gene bank collection or a wild population of plants is really akin to finding a needle in a haystack. And I would argue that plant phenotyping is always going to be a bottleneck in discovery of useful traits in crop wild relatives. To give an example, the four pictures you see here are another wild potato species, Solanum polyadenium. And from top to bottom, they're grown under increasing herbicide concentrations. Right, so you can see low or no herbicide concentrations, very happy plants at the top. At the bottom, the majority of the plants have been killed. So this plant obviously lacks herbicide tolerance. But in this study, I really wasn't looking for herbicide tolerance at all. I was trying to figure out how to grow these wild species in my late blight nursery in a way that didn't require hours and hours of hand weeding. Right, so something as mundane as how do you cultivate a wild species becomes a hurdle to discovery of useful traits, because if you can't do it on a large scale in a phenotype-driven approach, something like cultivation is a big issue. Understanding what late blight resistance or any resistance phenotype looks like in a plant that is morphologically and physiologically distinct from crop plants is a hurdle. Pathogen race specificity is always a problem in plant pathology. So if I'm challenging this with one particular race of Phytophthora infestans, is it actually resistant to other races? And of course, phenotyping is inherently resource intensive. So space, labor, funding all represent hurdles for the application of phenotype-driven approaches. So if phenotyping is a bottleneck for the use of crop wild relatives, what's the alternative? And in the last 5 or 7 years, my group has been working towards utilizing our knowledge of gene function and advances in DNA sequencing technology to develop genotype-driven approaches to inform phenotyping for trait discovery. And the work that I'm going to share with you today is work that's happening in the Rosaceae, and you'll see why in just a moment. Now, there are probably people in this room that know this way better than I do, but in order to make sense of the project that I'm going to share with you, I want to start with a little primer on how plants recognize and respond to pathogens. And plants really have very sophisticated surveillance systems. There are multiple different strategies that plants use, but one of the predominant ways that plants recognize and respond to pathogens are through genes that we collectively call resistance genes, or R genes. And R genes, of course, encode R proteins. And regardless of the plant species or the pathogen class against which these genes act, the proteins that they encode share certain architectures. And the predominant superfamily of R genes encode an NB arc, or nucleotide binding site domain, and a leucine-rich repeat, or an LRR. So NB LRR superfamily is really very prominent in terms of plant recognition of pathogens. Now, for the purposes of this talk, I want you to remember that there are two flavors of NB LRR proteins. They both have NB and LRR domains, but they differ in the N-termini. So they're TIR domain here on the bottom, and a coiled coil or non-TIR type. So these are two distinct flavors of NB LRR genes or NB LRR proteins. And we know that they have monophyletic lineages, and that'll become important in just a moment. Now, in the plant, NB LRR proteins play the role of cellular sentinels and switches. So these are constitutively expressed at low levels, and the LRR in particular is involved in protein-protein binding. And through protein-protein binding, the LRR detects directly or indirectly the presence of the pathogen. When that happens, so that's the role of sentinel. And when that happens, it induces a protein conformational change that allows the NB arc domain in combination with the N-terminal components to trigger a really elegant cascade of gene expression that results in the phenotype that we see as resistance. So the LRR plays the role of sentinel, the NB plays the role of switches. Now, in addition to a pretty robust understanding of how plants recognize and respond to pathogens, we're also living in an age of really significant advances in DNA sequencing technology. This is a timeline showing whole genome sequencing in plants. And the first plant to be the genome of which was fully sequenced is Arabidopsis back in 2000, followed very quickly by rice. And for a long time, these were our model monocot and dicot species. But if you look out here at 2017, the rate of discovery, the rate of generating whole genome sequence in plants is really a lot more robust. And we've got something like 250 plant genomes that have been fully sequenced at this point. Now, this project that I'm going to share with you focuses in the Rosaceae, and some of the key players in terms of whole genome sequence are the Malus domestica, or apple genome, that was released first in 2010. Fragaria vesca, this is a relative of strawberry. It's a diploid species called, referred to colloquially as the woodland strawberry. And that was sequenced in 2011. And then Prunus persica, or the peach, was sequenced in 2013. So these were the first three of several genomes that were sequenced in the Rosaceae. Today, we've got a total of 12 rosaceous genomes that are publicly available. Rose is coming out very soon this fall, and I can't wait to get my hands on it, because the rose genome, even though the family's called the Rosaceae, named for rose, rose has not been sequenced, so it's not amongst the 12 that we're studying in this particular study, at least yet. So the availability of genome resources for Rosaceae made this a really good candidate for the work that we're pursuing. And as we have worked towards trait discovery, disease resistance discovery in particular, over the years, it's really been interesting to me that comparative genomics approaches have been rarely employed for study of disease resistance. So comparative genomics, are taking knowledge from one species and applying it to another, really has revolutionized many, many aspects of the plant sciences, but not disease resistance, and the NB-LRR genes in particular. There are many reasons why that's the case. First of all, plant genomes have many NB-LRR genes. This is a graph that shows the 12 rosaceous species that we're studying in this particular study. And I'm just plotting the number of NB-LRR genes that we found. So way over here on your left is the black raspberry genome. This has 43 NB-LRR genes. That's ridiculously small. Cucumber, papaya have maybe similar numbers, but most plants have many dozens or several hundred R genes. So not a lot of genes in rubus. In contrast, apple, shown here on the right, 663 NB-LRR genes. So there are a lot of R genes in plant genomes, and the plant genomes in the Rosaceae really vary significantly in terms of the number of our genes present. So that creates a problem for comparative approaches. Additionally, NBLR genes evolve very quickly. And this is a slide that my colleague Nevin Young provided to me. So Nevin, for many years, has worked in a model legume known as Medicago truncatula. And his team sequenced a very large number of accessions of Medicago truncatula. And this graph summarizes diversity of multi-gene families across those various genomes. The x-axis is a measure of synonymous substitutions across these gene families, and the y-axis is a measure of non-synonymous changes. So anything that's down in this quadrant is relatively slow evolving, and anything up here is very rapidly evolving. And if you look at these three individuals, these are domains common in NBLR genes. So we've got the NB-ARC, the leucine-rich repeat, and the TIR domain. So those are amongst the most rapidly evolving components of plant genomes. This particular gene is late nodulin, not an NBLR gene, but it is a protein that's involved in mediating legume rhizobium interactions. So in general, genes that are involved in plant microbe interactions tend to evolve very rapidly. That makes sense from a biological perspective because we know that microbes are small, their population numbers are much larger than we'd see in plant communities, for example. And we know that the life cycle allows them to adapt very quickly relative to plants. So in order to compensate that, the plants have figured out how to evolve our genes very rapidly to keep pace with the microbial populations. So the fact that these genes evolve really quickly presents another hurdle for applying comparative genomics approaches. Now, I've talked already about the general structure, the conserved architecture of our proteins. And of course, genes reside on a chromosome somewhere. But it's relatively rare to find an NBLR gene that exists as a single entity on a chromosome. Much more often, they occur in clusters of functionally divergent but sequence-related copies. And the fact that they're clustered within the genome is really important in terms of their evolution because unequal crossing over within individual genes or within that cluster can really lead to very rapid changes in the overall structure of these clusters. So these clusters can expand and contract, and you see a driver of really rapid allelic diversification as a result of this clustering. So this, in particular, obscures one-to-one orthological relationships. So that's sort of a hallmark of comparative genomics. You need to know that the genes you're comparing have a common evolutionary origin. And this in particular represents a really significant complexity in terms of applying comparative genomics approaches. This graphic actually shows what gene clustering, NBLR gene clustering, looks like in an actual plant genome. So this is Prunus persica, or the peach genome. There are 8 chromosomes, and they're arrayed end to end. Each bar that you see here is an NBLR gene that we've identified in our study. And you can see a really strong tendency for these genes to cluster throughout the genome. So this really is a very important mechanism for evolution of these genes. All right, so I want to take just a moment to summarize this idea of comparative genomics. So there are 3 main reasons why we haven't seen application of comparative genomics approaches for NBLR genes. First of all, there are many of them within the plant genome. They evolve very rapidly in response to microbes. And this idea of orthological relationships are really very rapidly obscured. What I'm here to tell you that I think there's a lot of potential for comparative genomics for NBLR genes. And I think the application of comparative genomics to this particular gene class can help us identify traits in gene bank collections for crop improvement. We're working in the Rosaceae. Probably don't need to introduce this family. It's horticulturally very significant. And it also, in addition to being horticulturally significant, has a very robust phylogeny. So we know that everything that we call Rosaceae today came from a common ancestor that lived about 100 million years ago. 59 million years ago, rose, raspberry, and strawberry diverged from a common ancestor. 52 million years ago, the pomes, so apple and pear, diverged from the stone crops, stone fruits. 32 million years ago, apple and pear diverged from a common ancestor. And we know that all the stone fruits today, the cherries, the peaches, the plums, have a common evolutionary origin that is more recent than 32 million years ago, but we don't know exactly when that was. So it's important that we've got really robust genome resources for Rosaceae. We've got a well-established phylogeny, but this phylogeny also has a really significant chronological component. And so this combination of traits allows us to ask and answer some really sophisticated questions about trait evolution. And of course, we're focusing here on disease resistance. So for the last several years, we've been working in this system to identify ways of essentially a strategy and a computer pipeline that allows us to make meaningful comparisons across species. This is Dr. Leon Van Eck. He's a postdoc working in my group, and he's actually been responsible for much of what I'm going to share with you today. Now, key to our approach to comparative genomics is recognizing that these gene clusters, while very important biologically, represent a degree of phylogenetic redundancy. So our goal is to look at ancient relationships of our genes and understand how speciation events in the family have influenced the R gene component in genomes. And the clusters that we see today, we know are relatively recent in origin, and we know that they have a common evolutionary origin. So all of these genes in a cluster today could be traced back to a single copy. So from a phylogenetic analytical perspective, they're really redundant, and we've opted to collapse these into a single composite sequence that represents that group. So we demonstrated through a series of empirical studies that an 80% protein identity level in the NB-ARC domain in particular is the correct definition of this diversity bin. And I'm happy to talk about why we ended up with that number. But this diversity bin, as we think of it, in my mind is akin to a species, right? So we all understand that species is, number one, an artificial construct, but that a species is a meaningful, biologically meaningful entity that represents a series of genotypes that are genetically diverse. In the same way, this composite group, this diversity bin, represents a set of our genes that are genetically diverse, but this still has biological meaning, as you'll see in a moment. And we've also created a standardized terminology to go along with this. So rather than calling these composite groups, we refer to these as Rosaceae groups, and that stands for Rosaceae, our genes at 80% identity, and we can number these sequentially. So as a community, this becomes a resource for dialogue about the evolution of these genes. So Rosaceae groups. So in the Rosaceae, we've got 12 different species. Our computer pipeline identified a total of about 2,500 R genes. One of the key things that we're doing in this study is applying a common set of rules across all 12 species. So the numbers that we identify in a particular species sometimes varies from what's published in the literature. That's because our definition, our stringent rules are a little different than what we see different communities applying. But what we call in our gene, in apple, really corresponds to exactly the same definition in peach or strawberry in this particular case. So these 2,500 R genes could be collapsed into 651 Rosaceae groups. This is still a large number, but it's small enough that we can start to visualize data in new ways. And a lot of this really is about data visualization and observing these evolutionary patterns. So one of the things that we've done in this study is to create phylogenies. This is a maximum likelihood tree generated by our pipeline from these 651 Rosaceae groups. So each of the branch tips here are these diversity bins that represent multiple different DNA sequences. Now, this tree can be annotated with all kinds of information. So here I'm layering on information about the representation of individual sequences across the various genomes in the Rosaceae. Now, I could show this as the 12 different species with 12 concentric circles. It gets really, really complicated very quickly. So instead, I've opted here to show this at a tribe level. So there are 4 tribes represented here in the Rosaceae: the Rubiae, the Potentillae, the Amygdalae, and the Pyriae. The Rubiae, in this case, is a single genome sequence of rubus, so black raspberry. The potentillae include strawberries. So we've got 6 different Fragaria species in this study. Amygdalae are 2 different prunus species, and the Pyrrheae are apple and pear. So this visualization allows us to see certain trends that genes are represented differentially across these various genomes. We also, at a topographical level, we've defined 9 different clades reflected in this outer ring, also shown by the color of the tree itself. And these are labeled clade A through clade I. And importantly, we can demonstrate that this tree, the topology of this tree, points to these 2 broad, evolutionarily distinct lineages of NBLR genes. So I mentioned the TIR and non-TIR types. So in fact, we see that same evolutionary pattern reflected in the structure of this tree. So that serves almost as an internal control, telling us that the pattern that we're seeing in this approach has biological relevancy. So a lot of time went into this simple diagram, but now that we've got this, we can start to make certain evolutionary observations about the patterns of diversification of these R genes across the various species. So I'm going to list 4 evolutionary observations coming out of this study. The first is that R gene lineages are predominantly of ancient origin. And what this tells us is that this magical plant that lived 100 million years ago that gave rise to everything we call Rosaceae today already had the remnants of those 9 R gene clades back 100 million years ago. And of course, as that plant diverged into distinct species, the plant physiology changed, the geographic representation of the plants changed, and the pathogens that it encountered changed. So we see adaptation of the R genes throughout speciation events. But almost all of the R genes that we see today can trace their lineage to a common ancestor that lived 100 million years ago. Secondly, we see from our analyses that everything is everywhere except when it's not. And by that, I mean that the 9 clades are predominantly, the 9 R gene clades are predominantly represented across the 12 genomes. There are a few exceptions. I want to highlight the exceptions because I think those are really the interesting case. I'm going to show this a couple different ways. It's difficult to represent this data, but I'll do the best I can. I'll show it in a couple different ways. So this complicated figure is best read from right to left. The right column here represents the 651 Rosaceae lineages that we've created. And we've broken these into the TIR and the non-TIR types. Next, we've mapped the representation of those lineages back onto the individual clades, so those A through I that we've defined. Finally, we've mapped the presence of the clades onto the 12 genomes that are analyzed in this study. Now, if you have really good eyes and you really stare at this a long time, there are a couple things that'll jump out at you. First of all, clade F right here, really, really tiny. And in fact, there's a single Rosaceae sequence that represents clade F in this particular study. Clade F is only found in apple and pear, so the pome fruits. It's absent in the prunus, it's absent in strawberries and in raspberry. So that tells us that that particular clade is probably of recent origin. So most of these are really ancient, 100 million years ago. This one had to have evolved sometime in the last 32 million years or so. Also, if you study this for a long time, you'll see that clade A, well represented across the Rosaceae, but missing in the 2 prunus species. So we hypothesized that this might have been lost sometime within the last 39 million years ago. Now, I can show this in a different way on this tree you've already seen. Clade F is this thin blue line right here, present only in apple and pear and missing in the other species. And clade A is missing in the 2 prunus genomes that we've studied. So that must have been a recent loss, at least no more than 52 million years ago. Now these are really hypotheses. You know, our data set, while we've got 12 genomes, it's ultimately limited. And there are both technical and biological reasons that we might see this pattern. So this really warrants more analysis. We'll be analyzing more genomes. But these are really interesting patterns that we can only see through comparative approaches. Now, another exception to the everything is everywhere rule comes from Fragaria species. So we were fortunate to have access to 6 different genome sequences of Fragaria species. And the 6 species here are shown as columns in this table. And the 9 R-gene clades that we've defined are rows. Now, there are a couple types of data that are represented here. The size of the circle indicates the relative number of individual DNA sequences per clade per species. A related measure is the color, which is the number of rows or 80 groups per clade per species. Really, the thing you need to know to understand this is if you see a circle, that means there's an R gene from that clade in that particular plant genome. And the bigger the circle, the darker red the circle, the more individual sequences are present. So a couple things jump out immediately. Clade F, that's that tiny little clade that we see only in apple and pear, completely missing in our Fragaria species. But this species is Fragaria vesca. And this was one of it was the very first sequence for Fragaria species. And Fragaria vesca has really been touted as a model system for analyses in the Rosaceae for many different applications. So it's really sort of disturbing to me that we found no evidence of clade A, D, or F, E in this particular plant genome. Again, there could be technical reasons, there could be biological reasons. Maybe if we sequence a different genotype of Fragaria vesca, those sequences would be present. But I think this is sort of a wake-up call, I guess, for overuse of model systems in studying plant genomes. So what we really need are lots of plant genome sequences from lots of different individual genotypes within each species. So a third evolutionary observation from the study is that patterns of R gene homology reflect species phylogeny. And I'll explain what this means in a moment. But this really is justification for a comparative approach for R genes. So we've remembered that in this study, we've collapsed the phylogenetically redundant diversity in these R gene clusters into a single composite sequence. Importantly, these sequences, while defined at an 80% identity level for the MD protein, we have not constrained species representation. So in some cases, these groups comprise sequences from only one species. In some cases, they might contain sequences from multiple different species. Now, I want to clarify that I'm talking about the representation within individual rows or 80 groups. So a broad clade could be represented across the Rosaceae. But for example, and we'll talk about this in just a moment, these sequences shown right here are found only in apple. Right? So that's an example of a rows or 80 group that comprises sequences from one species, even though the clade overall is represented across the Rosaceae. So if we only look at representation within the Rosaceae, we see that the majority of individual rows or 80 groups comprise sequences that come only from one tribe, so closely related species. And that's represented by the numbers in these circles. And this makes good sense, right? This is consistent with this model of rapid evolution of R genes to adapt to pathogen pressures. But when we see rows or 80 lineages comprising sequences from more than one tribe, the more closely related the tribe is, the more common shared individual sequences are. So for example, we see the stone fruits and the pomes sharing 17 rows or 80 groups in common, and Fragaria species and Rubus species sharing 4. That's in contrast to the more distantly related tribes. So the more distantly related you are, the less likely you are to share sequences in common. That makes good sense. That's really what phylogeny is built on. But this more or less serves as a justification for comparative genomics approaches. So this really works for our genes as well as for other genes. Now this graph also shows something that I find just fascinating and perplexing, and I would love to hear your thoughts on this. We always think about our genes as evolving very rapidly. I showed you the graph. I've used that terminology multiple times. But there are certain R genes that we've found in the study that are very slow evolving. And in particular, 4 rows or 80 groups comprise sequences from all the tribes. So over 100 million years, these genes, the protein sequences, have not diverged by more than 20%. that does not match a pattern of responding to changing pathogen pressures. So one of the key questions that comes to my mind is, what are these sequences doing? There are a few models that we've got in mind. They could be involved in non-host resistance, and maybe they're so good at what they do, they've been conserved in the Rosaceae, and they're defending against a would-be pathogen that we don't even think of as a pathogen for the Rosaceae. So that's one possibility. They could be engaged in mediating positive plant microbe interactions. Or maybe they have entirely different functions. They could be involved, for example, in environmental stresses. So there's some key experiments we can think of, look at their transcription patterns, upregulate them, knock them out. But really, what do you challenge these plants with? If you knock one of these genes out, what do you challenge with? So I'd love to hear your thoughts on exactly how we can tackle what these things are doing. But this really, I think, illustrates the power of comparative genomics. If we were looking at the genome sequence of apple, certainly those gene sequences are present there. We would have no idea that they're highly conserved. So through this comparative approach, we begin to see that understanding these patterns of diversification actually can tie back to putative biological function. That's really what we want to leverage in this study. Now, the fourth and final evolutionary observation I want to share with you, I think, is the most exciting. And that is that species differ in patterns of our gene diversification. And this really provides clue to how to use this to mine those gene bank collections. So we've seen this tree before. I've already pointed out that clade E is well represented across all of the species in this study. But within the clade, there are certain subclades that show very different patterns. So this subclade that I'm highlighting is a good example of diversification that's happened only in the pome fruits, only in apple or in pear. So very, very well represented in apple and pear, but this subclade is absent in these other species. To me, this means something. There's an evolutionary reason why this has happened. And I would predict that that has some biological function that's important to apple and pear, but irrelevant to the other plant species. Now, there's another layer of data that I want to layer on here, and we'll talk in more detail about this. This outer circle that you see here is a heat map that shows the representation, shows the composition of the individual Rosaceae groups. So remember, the Rosaceae groups are the branch tips here. So this actually shows you how many individual DNA sequences went into defining that R gene, that the Rosaceae group. Dark color indicates 2 sequences, which is the minimum definition. And in this study, the largest Rosaceae group has more than 60 sequences, and that's shown as white. So within this subclade, for example, we see a multitude of colors, orange being sort of in the 20 to 40 individual DNA sequences going into that group. But if you look across the tree, you see many examples of this sort of pattern of evolution. So apple and pear really prefer this clade, telling us that there's something happening in a functional evolutionary way. In contrast, strawberry loves this particular clade. So understanding what these patterns are, I think, is really key to linking DNA sequence to putative function, and then ultimately mining these for the improvement of crops. All right, so the 4 evolutionary observations, just to conclude this piece. Our gene lineages are of ancient origin. Everything is everywhere except when it's not. Patterns of our gene homology reflect species phylogeny, and species differ in patterns of our gene diversification. So that was all sort of observational science. And the next phase, I want to tell you about a couple hypotheses that are emerging from here that really only happen because we're looking across species. So one of the areas I've really been interested in for a long time is an answer to this question: has agriculture escalated the molecular arms race between plants and pathogens? There's really an elegant literature out there that documents that large-scale monocultures practiced in modern agriculture has had significant impact on the effector suites of pathogen populations. We've seen that in multiple different pathosystems. I think it's perfectly logical that we would see the same pattern in plants. It'd look a little bit different. So in plants, what we think is happening is that pathogen pressures are driving plant genomes to diversify specific alleles. And as we talked about in terms of clustering of these genes, one of the key drivers for allelic diversification is expansion of our gene clusters. So thinking about evolutionary patterns where individual species have lots of our genes and high allelic diversity in certain clades or subclades on our tree, we think is an evolutionary indicator that pathogen pressures are causing the plant to diversify in those genomic regions. Now, to test this, we've turned to the apple scab pathosystem. Now, apple scab is aptly named. It causes scab on apple. It's caused by the fungus Venturiae inequalis. And it has, for a very long time, been a target of apple breeding efforts in particular. So there have been a total of 20 R genes, or resistance genes, defined at a phenotypic level in the apple scab pathosystem. These are labeled RVI1 through RVI20. I think 2 of these have been cloned. They are, in fact, NBLRR genes. The rest of them have been mapped to varying degrees and are useful for breeding applications, but we don't actually know what the underlying gene is. So in this study, we culled the literature and put the associated molecular markers back onto the apple reference genome, and we could find likely NBLR candidates for 15 of these genes. And when we looked at those 15 genes, we hypothesized the apple scab genes should occupy these large species-specific, highly diversified Rosaceae clades. And in fact, I'm showing it only for 3 of these, RVI 4, 15, and 1, the candidates for those genes reside in that highly diversified subclade in clade E that I described. And in fact, each of these comes from a Rosaceae group that contains many different DNA sequences. So this pattern of allelic diversification, I think of it as really iterative evolution. Perhaps apple hit on some gene sequence that worked really well against Venturiae inequalis, and then evolutionary dynamics created more allelic diversity at that locus, and some of that allelic diversity works against Venturiae inequalis and some of it doesn't. But this idea of reinventing functional genes seems to be a pattern of evolution that plants are adapting. And it's reflected very nicely in the overall topology of our tree. Now, looking across our 15 candidate genes, we found really strong statistical support for this idea that important agricultural genes reside in these highly diversified clades. So that gives us a clue in how we can actually go after gene bank collection. So if we develop strategies for generating sequence data from gene bank collections at those loci, we suddenly have a way of recognizing which genotypes or species or populations have novel DNA sequence or protein level diversity. And that information then can inform phenotyping. So this used as a system can really speed up discovery of useful traits for plant breeding. And so this idea of mining is really where we're going. Now, the first step towards that we're pursuing actually in Rose, in the Rose black spot pathosystem. So Stan Hokinson's a professor at University of Minnesota. David Zlezak is at the University of Wisconsin River Falls. I've collaborated with these 2 guys for probably 10 years on Rose black spot issues. Stan's actually a breeder, and he's part of the USDA CAP called Rose Breed. And the focus of that is to develop molecular markers linked to useful disease resistance traits like black spot in rose. So in connection with that project, we've already generated large segregating populations, and we've done a lot of phenotyping data in SNP marker analyses. We want to leverage that data set and our Rosaceae system to identify perfect markers for marker-assisted breeding. And in this context, I'm defining a perfect marker as one that's actually derived from the causal gene. So the goal is to actually land on top of the allele that makes rose resistant to the black spot pathogen. And for this, we're adapting a DNA sequence capture approach called RenSeq. So this was pioneered by Jonathan Jones's group in, ironically, the potato late blight system. The idea is that a series of DNA sequence baits are created from known R gene sequences, and that's used then to pull down corresponding, not necessarily identical, but corresponding sequences from DNA solutions from, for example, gene bank collections. That gives us a pool of R gene enriched DNA that can be sequenced at large scale. So we're using this in Rose. We're creating DNA baits from what we've learned are the 9 clades in the Rosaceae. We've already created resistant susceptible bulks based on our Rose phenotyping that we've completed. Our goal is to create sequence libraries that have allele level resolution. We'll identify candidate polymorphisms and of course validate on the larger data set. So this really serves as a proof of concept that would be directly applicable to rose breeding, but I think could be really ported to almost any plant family, almost any gene bank collection. And so for example, downstream, we'd like to do this in the apple scab pathosystem or in potato late blight or in wheat rust. I think it has really a multitude of applications. So to wrap up, in this seminar, I've presented comparative genomics framework that we're calling the Rosaceae system. This is really what we've done is to define a set of rules for looking across plant families at the evolution of these genes. And we've created a computer pipeline that does that in a semi-automated fashion. In the Rosaceae, some of our evolutionary observations are that our gene lineages are at least 1 million years old. Everything is everywhere almost. Our gene distribution reflects phylogeny. And the most important is that species differ in patterns of our gene diversification. That is our entree into hypothesis testing, linking individuals, our gene sequences or our protein sequences, to putative biological function. And of course, the end goal here is to really effectively mine gene bank collections for crop improvement. Hopefully the genotyping will drive informed phenotyping that will sort of fast forward our process of plant improvement. And with that, if I've got time, I'm very happy to take any questions. Yes.
Are gene clusters due to an year of weekend mutation hotspot in the genome?
Yeah, so the question was, do our genes tend to cluster near recombination hotspots in plant genomes? And the answer is yes. So this idea of clustering is really important in terms of driving overall evolution and adaptation, creation of new alleles at these loci, but other factors like being in genome locations that are more prone to recombination is another key piece of that. They're often associated with transposable elements, which I think probably has a biological role. So there are many factors. We probably don't have a full, complete understanding of what's driving it, but your question's spot on. Yes.
So I want the issue with these conserved effectors. So do you consider that maybe they are guarding the very conserved plant products?
Are you talking specifically about the four?
The four very conserved.
Yeah. So great. Yeah. So the question referred to that small set of R genes that really has not evolved very rapidly over 100 million years. And the question was, are these R genes, are the cognate effectors highly conserved in pathogen populations? Oh, sorry.
They are guards of the highly conserved plant proteins.
So you're asking if the conserved R genes are guards of highly conserved plant proteins. Probably yes. Actually, I would even go so far to say that the cognate effector probably is highly conserved. And I think that you know, whether that is sort of a non-host, that's consistent with the idea that they might play a role in non-host resistance. I think it also could be consistent with the idea that they may be involved in mediating positive plant-microbe interactions. So I mean, I'm speculating at this point because we don't have any data about that, but probably I think that's a logical.
Wonderful.
Okay.
So you have this unique F-glide in these apples and pears. The F-glide.
Okay. Yes.
That is only in apples and pears, and there is some very different pathogen than attack only apples and pears and others. Or
yeah. So it's a great question. I wish I knew the answer. So the question is in that F clade, that tiny little clade, R gene clade that seems to only be represented in apple and pear. What's it doing? Is it corresponding to some unique pathogen that's only found in apple and pear? And I don't know, probably, but there are many unique pathogens of each of the species in the study. So it's a good guess, but I don't know how to narrow that down. Bill.
Do you have any hypothesis for the dramatic difference in numbers of our genes among the different?
Okay, so Bill, Yeah, it's a great idea. Bill's asking why do the number of R genes in Rosaceae genomes vary from 43 in rubus to more than 600 in apple? I don't know, but I think it actually means something. So some of our let me give two schools of thought on this. There's this idea out in the literature that woody plants should have more R genes because they're longer lived. I have traced this back to the poplar genome sequence. It seems to be just sort of put out there as a hypothesis. I've seen it in multiple different papers. I see no evidence of it. And I don't understand how a woody plant says, oh yeah, I need lots of R genes. I don't think that's the driver. What I think is actually happening is relates to the agriculture intensification of these things. So more highly, intensively farmed plants, things that have a longer history of cultivation, things that are a broader worldwide distribution, I think should have more R genes, especially compared to the wild relatives. And even though we've got 12 genomes in the study, we don't really have that many wild species. So one thing I'd like to do with our pipeline is do sort of a meta-analysis of the 251 plant genomes that we do have, and start to look at life history of the plant, cultivation history, the agricultural significance of it, whether it's woody or not woody, and see what the correlation is with the number of R genes. So I don't know. It's a great question. I'm a biologist. I think there has to be a reason for it, but I don't know what it is yet.
Last question.
Sorry, so what ways does agriculture facilitate the diversification of R genes? Is it artificial selection? Is it...
I'm sorry, you're going to have to speak up.
Oh, sorry. In what ways does agriculture facilitate the diversification of R genes? Is it artificial selection? Is it just one type of pathogen?
Right. So this idea, the question is relates to agricultural intensification. Why? What's the driver? How is that influencing the putative change in R gene numbers? I think they're both intentional selection, unintentional. So agriculture, as we know, it's 10,000 years old. So somebody had a great idea 10,000 years ago to actually plant some of the seeds that she went out and harvested. That act alone, the domestication process, put selection on the plants, right? So plants that don't shatter, plants that overwinter, plants that taste good, et cetera. So there was an inherent selection that went on during that domestication process. I think probably people 10,000 years ago didn't want to eat scabby apples any more than we do today. So they probably ate the ones that looked good. It wasn't intentional selection, but it was selection. Of course, in more modern times, breeding, we're intentionally putting that selective pressure on there. So I think it's probably a combination. One of the key things that has come out of our study, though, is it's really changed the way I think about evolution. I've always thought of evolution as happening over millions of years. Now I'm really curious, our genes, like, are they even generation to generation? Are we seeing changes in these clusters? And I think that'd be really a really cool set of experiments to do. And with whole genome sequencing becoming more accessible, it's something we probably can do in the not too distant future.
All right, let's thank Jim for a great talk.
Thank you very much.
This has been a production of Cornell University, on the web at cornell.edu.